Physical, chemical and microbiological or cellular barriers limit entry of pathogens. There are several portals of entry by which pathogen can enter into the body: the intact skin and the mucous membrane, which extend over the body surface or into the body cavity, and their indigenous microbiota are defenses providing barrier to infection at this exposed site. Physical barrier: the keratin in the skin, outer layer, the epidermis, form a tough and impenetrable barrier to infection. In addition, thin outer epidermis is constantly being slugged off, and with it goes any attached microorganism. The skin also is a poor source of nutrition for invading microbes, and from the viewpoint of a pathogen, the low water content of skin present a variable desert. Unless microbes can penetrate the skin barrier, disease is rare, although some dermatophytes can lead to disease like athlete foot. Cut or abrasion are a fact of everyday life. These breaches to skin may allow microbe to enter blood. Bite from infected arthropods acting as hypodermic needle may inject the pathogen responsible for such disease as West Nile fever, malaria or plague. Other means of penetrating this mechanical barrier include puncture wound, tooth extraction, burns, shaving knives and injections. Possible penetration of skin barrier is patrolled by dendritic cell called Langerhans cell in the skin tissue. If a pathogen is detected by this cell, they phagocyte the pathogen and migrate to a regional lymph node where they induce and acquire immune response. The dendritic cell also secretes cytokines, which influence both innate and acquired immune response. The mucous membrane or mucosa are the moist epithelial lining of digestive, urogenital and respiratory tract. And urogenital. Because these exposed sites represent potential portal of entry. The mucous membrane provides resistance to microbial invasion. For example, cells of mucous membrane that line the respiratory passageway secrete mucus, a sticky secretion of glycoprotein that trap heavy particle and microorganism. The epithelial cilia then move the microbes laden mucus up to the throat where the material is swallowed or coughed out. Chemical barrier. Several host chemicals also provide an antimicrobial defense to infection by inhibiting microbial growth or viral replication. The mucous membrane, mucosa, are the moist epithelial lining of digestive, urogenital and respiratory tract. Because these exposed sites represent potential portal of entry. The mucous membrane provides resistance to microbial invasion. For example, cells of the mucous membrane that line the respiratory passageway secrete mucus, a sticky secretion of glycoprotein that trap heavy particle and microorganism. The epithelial cilia then move the microbes laden mucus up to the throat where the material is swallowed or coughed out. Chemical barrier. Several host chemicals also provide an antimicrobial defense to infection by inhibiting microbial growth or viral replication. Resistance in vaginal tract. is enhanced by low pH. This develops when lactobacillus species in microbiota break down glycogen to various acids. In the urinary tract, the slightly acidic pH of urine discourages pathogen growth and the flow of urine flushes the microbes away. A natural barrier to the gastrointestinal tract is provided by stomach acid. With a pH approximately 2, destroy most pathogen. However, there can be notable survivors including polio and hepatitis A viruses, the typhoid and tubercle bacilli and Helicobacter pylori, a major cause of peptic ulcer. Bile from the gallbladder enter the system at the duodenum and serve as an inhibitory substances. In addition, duodenal enzyme hydrolyze the protein, carbohydrate, lipid and other large molecule of microorganism. The chemical barrier also consists of numerous small antimicrobial peptides called defensin, which are found in various secretions throughout the body. They are produced by phagocytic cell as well as epithelial cell of the respiratory, gastrointestinal and urogenital tract. The sebum on skin surface, for example, produce at least 12 defensin. Some defensin are continually produced while others are induced by microbial product. They are active against many gram-negative and gram-positive bacterial cells, fungi and viruses such as HIV. Many of the defensins damage membrane, killing the pathogen through cell lysis. In somewhat similar manner, lysozyme is a chemical inhibitor found in human tears, mucus and saliva. The enzyme disrupts the cell wall of gram-positive bacterial cell by weakening the peptidoglycan. Osmotic lysis results in cell death. Other soluble proteins such as complement component and interferons are important physiological barrier in innate immunity. Cellular barrier. The blood monocyte tissue, macrophages and neutrophil are key cellular defenses of innate immunity. This cell can carry out phagocytosis and trigger inflammatory responses to destroy invading pathogen. Besides this leukocyte, defensive barrier also include the normal microbiota of the body surface. These indigenous non-pathogenic microbes form a cellular barrier by outcompeting pathogen for nutrient and attachment site on the skin and mucosa.